Hello, this is Ben Stanislav. For today's presentation, I chose a paper published by the Merzenich lab in 1991 that's looking at plasticity following artificial syndactyly. In terms of information already known, previous papers by Merzenich noted that the somatosensory reorganization can take place in adult monkeys, that this reorganization can cross previously discontinuous digit boundaries, and that receptive fields can change size and location. This all points to the concept that the dynamic representation in the somatosensory cortex based on input selection. But the question remains, how are these inputs selected? This paper tests the hypothesis that selection is based on temporal synchronization of inputs. Thus, the idea is that digits are represented in discrete cortical zones, not because of the anatomy of input projections, but rather because skin inputs from adjacent fingers lack temporal synchronization. Figure 1 provides a visual representation of what the artificial syndactyly that was produced in this experiment looks like. For this experiment, the skin of digits 3 and 4 were surgically conjoined in 3 monkeys to try to create conditions for temporal coactivation. Basically, every time they use digit 3, they will also be using digit 4 because they are conjoined. After 14, 25, or 33 weeks, the cortical representation of these digits in area 3B of the somatosensory cortex was mapped using microelectrodes, and in 2 of these cases, following the first mapping of the syndactyly was surgically released to create the original separated digits, and a follow-up microelectrode mapping was done. So in figure 2, we see what it would look like in a normal monkey. And what we can clearly see here is that in normal representations of the digits, they are discontinuous, meaning the inputs for 3 are separated from the inputs from 4. And what we see here is this discontinuous representation with abrupt borders between the adjacent digits is corresponding to receptive fields that jump from the individual digits. Now, moving along to those 3 monkeys that had the artificial syndactyly, what we see here are they were either mapped at 5.5, 7.5, or 3 months after the syndactyly, and those are respective time periods. So following the syndactyly surgery, would cortical representation of the digits remain discontinuous as we saw with the normal monkey, or would an area receiving input from both digits now be observed? And what we see here is that the normal discontinuity was replaced by a zone, or rather that shaded region on the cortical maps, a zone receiving inputs from both digits 3 and 4. In figure 4, we see the area 3B with microelectrode penetrations numbered. These microelectrode penetrations correspond to receptive fields, and this is for the first monkey. And this raises the question of will the cortical zones receiving inputs from both digits 3 and 4 have corresponding receptive fields that span the skin of fingers 3 and 4, or will there be duplicate receptive fields, one on each digit giving input to the same cortical zone? And what we see here is that largely the receptive fields are spanning the line of syndactyly to cover parts of digits 3 and 4. These receptive fields are corresponding to the shaded cortical zone, which tells us that the receptive fields can change and cross previous boundaries. There is some evidence also for there being duplicate receptive fields, especially at the tips of the fingers. And this is the case because in the syndactyly, the tips of the fingers are not fully fused, but we still get that same kind of coactivation. And so we still see that they are activating the same cortical area despite not being continuous. So the last thing that they did in this paper was following the first mapping of the syndactyly, they released it in 2 of the monkeys and then followed up by mapping it again. And the question they wanted to ask was, would the receptive fields remain functional given that the input from both were now separated digits, or would the receptive fields be denervated? And they saw no significant change in the size or location of receptive fields on the hand, meaning the overall topography was unchanged directly after release of the syndactyly. And this helps to rule out the possibility of receptive field changes from peripheral sprouting, which would have otherwise been severed when the syndactyly was released. Now, This experiment seems to demonstrate that the cortical representation in the area 3B is the result of a dynamic process based on temporal synchronization of inputs rather than static anatomical projections. And the take-home message is that the somatosensory cortical representation reflects distribution of coincident or near coincident temporal patterns among the afferent inputs, which supports the original hypothesis that the temporal synchronization plays a key role in the topography of the somatosensory cortex.